It's my pleasure to introduce Robin Wolfe Scheffler, in his bylines and book covers. He's associate professor in the program of Science, Technology, and Society at MIT. And his works really range across, I think at this point, all of 20th century and into 21st century biology and biotechnology, because I remember early work on physiology, human physiology, and then, of course, a book that I think is extremely interesting to people at Cold Spring Harbor and anyone with interest in history of the kind of research that's happened at CSHL. That was Robin's book on cancer viruses called A Contagious Cause. And he's working on a new project now that we're going to hear about. That's already I think, acclaimed, even in progress project about the history of biotech in Greater Boston. Robin, thanks for being here, and take it away.
Thank you. Yeah, thank you so much for having me, and thank you, of course, to Alistair and Mila and Tom and Trisha for sort of arranging this and giving me the opportunity to speak with all of you about some of the questions of exploring my current project. But I also want to spend some time highlighting explicitly how the Brenner Fellowship and the archives at Cold Spring Harbor Laboratory really have given me a unique perspective on some of the really big questions I'm wrestling with in the history of biology, biotechnology, and American society, which at whatever time it is the sort of the space I like living as a scholar. So I'm going to share my screen with you now. So at least for those of you who don't know me, you've had a chance to see my face before I disappear into a small, tiny corner of your screen. So without further ado, and I'll come back to this. Okay. There we go. Can someone unmute themselves and tell me that you can see a nice big blue screen?
We are seeing your title slide, Robin. Thanks.
Perfect. That's wonderful. Yeah. So I actually will say it was kind of you, Alistair, to mention my first book. I started working on this indirectly almost a decade ago, as I thought I was going to write a very quick chapter in my first book about how the war on cancer interacted with the modern American biotechnology industry. I spent about 9 months trying to write that chapter, and it got longer and longer and longer. And at a certain point, I realized that I wasn't actually trying to finish a first book. I was starting a second book. And researching that second book, which is my effort to sort of make sense of the role that the modern biotechnology industry plays at American society, which of course is a sizable one in many ways. Many of us carry its products in our arms and bloodstreams right now, and we interact with it in many of the ways that biology is central to our society. But really how that industry developed and the particular ways it interacts with society. And some of these are not ways that historians of biology necessarily always think. And in particular, the real challenge I'm trying to do in my project now is figure out how to tell the history of biotechnology through the history of a city. And I had originally thought I was going to do this, the history of the San Francisco Bay Area, which is sort of the heartland of my birth. But I ended up at MIT, and I started realizing, as much as it pained me as a Californian to admit it, that there was quite a bit of very interesting biotechnology and biology happening around there. And indeed, it has given itself the self-assigned moniker Genetown, which is a wonderful, catchy title, was causing me any number of issues with a laboratory supply company trying to figure out the trademark issues behind my book. But I just put this up here. I don't need to engage in too much special pleading, but the sort of biotechnology cluster in what's called the Greater Boston area, because Boston is this patchwork of over 50 cities, is the most vibrant and densest cluster of biological, biomedical, and biotechnological research in the world. There are certain neighborhoods, such as those around MIT, which I'll return to, which contain, depending on how you reckon it, 10 to 15% of the modern American biotechnology industry. So looking at this area is both a great way of learning about the specific history of biotechnology in Boston, which is definitely worthy of a book or more in its own right, but it's also a wonderful way of thinking about the role that biotechnology has in American history writ large, in, of course, one of the cities most associated with characterizing, defining sort of the American vision, the American dream. So this cluster, however, from many other perspectives, just doesn't make sense. And this is sort of the problem that I wrestled with as a historian, is sort of trying to both explain the history of this cluster, but also try to make sense of how it came into being, despite the multiple reasons that people in the broader world of innovation studies, science and technology would tell you that it should not exist in its current form. And I'm just going to, some of these reasons may be familiar to all of you, but I'm going to sort of just outline them very broadly. For starters, if you want to mention sort of high technology, high science-based industries after the Second World War, you get places that look like this: Bell Laboratories in New Jersey, or Stanford Research Park, and sort of the Bay Area of California. These are places that, are, of course, are characterized by not being set within cities. They're characterized by pleasant suburban surroundings, as you can see, especially for Bell Labs, ample parking. This is what I later learned office developers call the fried egg model of office and laboratory design, which is the buildings all in the center and then a lot of parking around it. So these, of course, are the kinds of places that are constructed as science and technology research moves into these growing American suburbs. And this is based on a very particular set of assumptions about sort of who those people are working in laboratories, which is sort of often a male, white, highly educated person, often working in physics, who wants to raise a family. And the zones that we associate with these very sort of high-tech industry are sort of places such as Silicon Valley, or as in this cartoon, Route 128, which sort of shows the foreshortened view that Boston has of itself, where you sort of have that very dense cluster of businesses, and then eventually you have this very big leap across the Pacific Ocean to Japan. So this is where high technology was happening and cutting edge science was happening in the 1970s, which is right, of course, roughly when the modern biotechnology industry gets underway. And you might think that it would have found its home in suburban pharmaceutical research laboratories like this one in North Carolina, which is typical, of course, once again, of that idea of suburban scientific research and development. And to give away the punchline of the story, it does not. It takes root in places like East Cambridge, which is going to be the place where I'm going to be spending my time for this talk. And along the way, in trying to sort of make sense of this particular landscape, I really want to highlight how following one particular company makes sense of a lot of much larger historical and economic and scientific trends that sort of help put biotechnology in its place, if it were. So this is very briefly to sort of give you an introduction to the place and the problem. And for the rest of my talk, I'm going to sort of build up on that problem a little bit more, and then sort of talk about some of the things I found thanks to the Brenner Fellowship in the archives of Cold Spring Harbor, and how they've helped me think about and inspired me to sort of think about some of the dynamics of the biotechnology industry that I do think were not in any means obvious, absent having the opportunity to sort of get on the inside of its companies. So once again, why is a biotechnology cluster in Cambridge in particular such an extraordinary idea? For starters, it's definitely not suburban. This is East Cambridge, the area immediately to the north of MIT. In the early 1960s, you can see industrial storage tanks for chemicals. You can see canals. You can see factory buildings. What you can't see, of course, is a lot of places to have lunch, a lot of places to live. There is a lot of parking, but that's because a lot of buildings have just been torn down because the factories are being moved to the American South. So it does not really fit that model of a pleasant place to necessarily locate your budding biotechnology company. Moreover, the core technology of the modern biotechnology industry, recombinant DNA, is the topic of fierce controversy, as many of you probably know, in the mid-1970s. And just to put in a plug, I am, along with a few of the other people here, working on a volume for the Journal of the History of Biology that came out of a conference hosted at Cold Spring Harbor in the past fall on the 50th anniversary of the Asilomar meeting, which happened in a nice suburban sort of an oceanside retreat in California, but was taken up in the city of Cambridge, which became the first city in the nation to pass its own municipal regulation of scientific research with recombinant DNA, which was if you think about the scales we often talk about for where science takes place, we're familiar with talking about nations and individuals. You don't often think about individual cities trying to sort of steer the course of scientific research, but that's exactly what happened in Cambridge, which of course hosts Harvard and MIT, which are two very important institutions to sort of the modern history of molecular biology. And so in the mid-1970s, it very much appeared as if scientists were going to have to go into the streets to convince their fellow city members that it was all right to do work with this very exciting technology. Indeed, there's David Baltimore, who later, and Walter Gilbert, both of them who are future Nobel Prize. Actually, David Baltimore had already gotten his Nobel Prize in this photo, having to set up at a local farmers market and street fair in the summer of 1976 to explain their work to anyone who wanted to come and question them. And all of these concerns about the danger and safety of recombinant DNA, although they often are sort of dismissed as no longer being as relevant as they've been resolved by the scientific community by the early 1980s, hold on in the popular memory of cities such as Cambridge. So this is a cartoon that somebody sends one of their city councilors in the, I think, early 1980s, showing organisms escaping into the Charles River through a sewer. And these concerns for safety continue to reside at these sort of levels where historians of science don't often think about them. And even if the concern over the safety might be misplaced in our view, it maintains a very powerful force on where biotechnology companies hoping to use this technology can take place. Many of them just decide to go elsewhere. So Genentech, for example, doesn't set up in Berkeley or Stanford or San Francisco, which would be close to the three major biomedical research institutions in the Bay Area. It goes to South San Francisco, which is best known for, and it sort of puts itself right out in the water in an industrial area, definitely not a hotbed, until it shows up of biotechnology research. It's a problem as well for the Office of Technology Transfer at Cold Spring Harbor, where they're considering setting up their own biotechnology company. And they find out that they actually probably can't find a permitted laboratory for the production they want to do because their neighbors in Laurel Hollow won't zone for it and have their own concerns about safety. So these safety concerns sort of really do shadow the question of where biotechnology will take place. And that's what brings me to Biogen, which is a company that has a very unlikely journey, which begins to sort of explain this apparent paradox between the high level of regulation that Cambridge has, in particular, in the nation for biotechnology, juxtaposed with the fact that it does, in fact, end up becoming this incredibly dense cluster for the industry in the ensuing decades. And Biogen is founded not in Cambridge, not in San Francisco, not even in the United States, but in a meeting in Geneva, where a group of venture capitalists, a group of scientists from across North America and Europe gather together in a conference room and then later on for a nice dinner that includes Walter Gilbert, among others, to discuss the formation of a new company in 1978 dedicated to realizing the promise and potential of this new recombinant DNA technology. And there's so much I could say about the business history of Biogen that the papers at Cold Spring Harbor reveal. For those of you who have done any history of the biotechnology business, it is famously difficult to get on the inside of a company because many of these archives are either confidential or non-existent because the company itself has gone out of business. So the fact that Cold Spring Harbor contains this network of scientists, many of whom were advisors to biotechnology companies, and many of whom fortunately really saved a lot of their paperwork and donated it, is really a wonderful window into how the early industry worked that's very difficult to find in many other ways. And fortunately, Biogen's papers, as I'll show, give a really wonderful sense of at least how one company operated, which may not be exemplary, but it's also not extraordinary in terms of some of the issues that it faces in finding a home in Cambridge. So to return to Geneva in 1978, this group of scientists is familiar with the challenges of national regulation. Walter Gilbert, for example, ends up having to go to Porton Down in England, has a chemical weapons research lab, to conduct a number of key experiments, fearing that they would not be considered illegal in Cambridge. This is a group, therefore, that really does not want to find a single home for their research. They definitely don't want to find that home in Cambridge. So they originally envisioned Biogen as this international, decentralized company sponsoring research in laboratories in many, many different countries, technically headquartered at an island in the Caribbean to avoid taxes, and through this sort of multinational structure, avoiding any of the regulation that any one nation could impose upon it. However, putting this vision into practice rapidly begins to run up against many of the challenges of actually doing research in a way that will make a profit for its investors. the first sign of this is they eventually do decide to open a laboratory in the United States, and they choose Cambridge, working in conjunction with their main laboratory in Geneva. So when the Cambridge laboratory was opened, it was very much an offshoot. And looking over the shoulder of Biogen as they decided on Cambridge, out of all the sites they could have picked in the United States, really reveals a lot about how the early biotechnology industry works. So, for example, in the memos that I've found, they are looking into a U.S. laboratory, and they look at a number of places in sort of the Boston area, which they call the Cambridge, Massachusetts area. And there's a few interesting things in this small paragraph I've highlighted, which I'll point out. There is a relative lack of knowledge about the particular sites within Boston. They misspell both Somerville and Malden, which are the towns neighboring to Cambridge. They just say Route 128, which actually is a whole cluster of communities, and which is this is being written by the head of their European operations, so that's perhaps understandable. I wouldn't want to hazard my guess spelling any number of cities in Europe. But the really interesting thing is that the second to last sentence, which is the disadvantage of these locations, is that they are some distance from a university campus. And you can also see that Cambridge becomes desirable in the first sentence because there's a large amount of space within walking distance at MIT. And until I started looking at this, I had not appreciated the degree to which evidently biotechnologists like walking. And they like moving around on foot as opposed to by bicycle, or as we saw earlier, by car. And the planning stages of this new Cambridge laboratory are replete with, once again, the importance of this pedestrian level activity. So this is a map, for example, by the consultants considering different locations that they could use for siting within Cambridge. And you can see that within their reckoning, the two blue dots are MIT and Harvard as campuses. The green dot that they put on is the MIT Center for Cancer Research, and then the red dot is their one of the locations that they're considering. But this is very much a map with the main sort of direct walking ways highlighted within the grid. So it's already sort of a manifestation of how much they understand this as something that's going to be sort of fitting into a pedestrian scale environment. And this is why they ultimately decide it's worth dealing with the potential of regulation within Cambridge, rather than sort of carrying out the move of many other biotechnology companies of simply trying to find a community that's less likely to regulate. So this is so tempting, they're willing to sort of put up with any number of things in the early 1980s that were unknown in terms of what the regulatory process would look like. Indeed, they helped define the regulatory process within Cambridge for biotechnology, but they have a really good reason to do it, and that's because of this desire to be within walking distance of a major university center in molecular biology. And so thinking more about this is sort of a way of getting into what the work of molecular biology and biotechnology looks like at the time. This is Phil Sharp, another Biogen founder, in his MIT lab somewhat later. But of course, he embodies a number of the things about molecular biology in the early 1980s, which end up being really important, actually, this story, which is it's really painstaking work. You spend a lot of time doing stuff by hand. One postdoc working at MIT comments that they've gotten this long, expensive education. You more or less end up as a glorified technician, in the sense that PhDs have to do this very painstaking work, such as pipetting. It's not automated at all in these initial stages. You have these products that promise to speed the process up, but you also have quite a lot of humor. And I think if Jan Witowski is still on this presentation, I first learned of the Midnight Hustler through one of his publications. And if you read that, a lot of the jokes are about lab leaders, PIs, trying to get their graduate students to remain motivated. And some of these, a modified bondage belt to lash your graduate students to the bench, so they'll keep doing work. And this once again calls attention to exactly how difficult it is to motivate scientists to carry out the long hours of work you need to both generate either relevant scientific discoveries, or if you're a biotechnology company, to do the kind of research that's going to get you patents. And so it's not enough to convince the board members of Biogen to do this work. you've got to convince people like these, the members of Gilbert's lab, who are also joking about, here, got some momentum here. The members of Gilbert's lab, these are the people who will be the hands that sort of animate the early biotechnology industry. And it's about where they are willing to go and where they're willing, evidently, to walk. And so a lot of the focus on having a pedestrian scale site to put Biogen in is a spatial resolution of this their classic issue that we see in the rise of early biotechnology, which is the perceived conflict between working in industry and working in academia, and the different values imputed to both of them. There's a lot more I could say about this, but putting your biotechnology in an environment where you basically get to act like an extension of a campus is a very powerful way of solving this problem. And there's many references, not just for Biogen, but for many other companies, about the desire to be on the doorstep of another company, of a university campus and an academic community. The question, of course, is if that's enough. And the other thing that Biogen's papers reveal, which is really interesting, is exactly how precarious it is to run a biotechnology company, which is widely known, but the texture is really remarkable. It's one of the main issues, of course, that Biogen faces is standard ways of accounting for cost are all on the sort of the left-hand side of this profit and loss calculation that investors use. All of the research they're doing, as exciting as it might be, is counted as a loss, and they have no products. And these biotechnology companies won't for over a decade, so they need to raise a huge amount of money to build a product that isn't yet in existence, all where their main activity, their main value is basically just spending other people's money. So it's a hard thing to carry out. So even as they seek to expand, they sort of realize that it's very difficult to keep two different laboratories going. There is a renewed focus on actually manufacturing things, which is to say getting a large scale DNA permit for carrying out fermentation. And so over the course of the 80s, the Biogen Cambridge campus becomes larger and larger at the expense of the Geneva laboratories, despite the fact that Geneva laboratories were when this started, by far the center of the company's efforts. And you can see in the publications from members of the Geneva laboratory that they are very unhappy about this. They continue to ask for assurances that Biogen is actually an international company and not just a company run out of Cambridge. And although Biogen offers a certain number of assurances, the end of this story is that the Cambridge laboratories are sold in the Geneva laboratories are sold in 1987, and Biogen consolidates to Cambridge because of the desire to not only be close to a university campus, but another thing that we often talk about in the history of science, which is the desire to keep the people doing the research close to the people doing the manufacturing. Because it turns out it's very difficult to talk about the scientific work at a distance. And a company like Biogen, which at that point is struggling to get products to market, really wants to have everything in one place. So you have this amazing story of thinking you can do work on a decentralized national basis, collapsing down into one laboratory in one city, and that city is Cambridge. And I'm just going to conclude by sort of talking about then how this fits into sort of the urban history of America side, which is what makes this kind of a drawing possible. What gives Biogen the confidence it can put a plastic overlay over a map of Cambridge and just sort of pick a site? And the answer is, at the time, that part of Cambridge actually does look like that map. It's been leveled, it's flat. You can see there's two buildings that in the foreground of this picture. The one that's sort of off to the lower, near the intersection in the lower right, is the Whitehead Institute, which is a major center of molecular biology. And then if you sort of go to the left of the Whitehead and then up, and you see a blocky building, that's the Biogen building. And then, of course, around it is really just cleared land. This was land that had been cleared to build this hypothetical building, which was a NASA research facility, which you can see here in outline. Of course, it remains imaginary because for a number of very interesting political reasons, it was never actually constructed. And it left that huge amount of cleared land. And so Biogen and other companies showed up in the wake of that controversy that had sort of really roiled East Cambridge for quite a while in the 1960s. And they became part of sort of redeeming that land for more productive economic purposes. And the growth of biotechnology by the city of Cambridge goes from being a possible hazard to a possible means of economic security and salvation. And that is sort of the dominant frame you see. And it's once again all based on this claim to proximity. indeed, there are now real estate companies who will, laud the pedestrian level sort of proximity of this billion-dollar portfolio of real estate for biotechnology research in East Cambridge and other clusters in the United States. And you can really then see it in this concentration from the 80s to the 90s to sort of the near past. And that's really where this sort of intense clustering gets on. And I really would not have appreciated exactly how that worked absent the opportunity to sort of look through the Biogen slides. But I do want to end on sort of a note of caution without being too hagiographic. This is the Whitehead Institute. Once again, a similar picture of the demolished land that Biogen and other companies moved into. Which is that part of the additional thing I'm thinking about in this project is that this desolation, of course, is not a natural fact. It's created through a number of historical processes involving America and American cities. And it also presents, therefore, a really interesting way, given the close relationship between the need of biotechnology companies to be in particular city spaces to be successful. It gives a very different perspective, therefore, on how to write the history of biotechnology, not just as an independent industry that could be anywhere, but as an industry who ends up being deeply linked to the fortune of particular cities around the United States, including Cambridge and other sites within Boston. And that really is sort of where I am now. But the ability to go inside the Biogen files in particular has been really, really helpful for giving substance to what had just been a very strong suspicion a few years ago. So with that, thank you so much. And I look forward to your questions.
Thanks so much, Robin. Fantastic. Let me first give the opportunity to ask questions to everyone else in the audience. Once again, if you'd like to ask Robin a question, please use the reactions tab at the bottom of your screen, and then we'll ask you to unmute yourself. Robin, let me ask first while people are locating that reaction tab. So you emphasized the sort of claims at the outset, or the ambitions at the outset, of a biotechnology industry that would be based on this pedestrian access to institutions of research and higher education. Did you find any evidence of people reassessing that or questioning at any point whether it had been worth it? Basically, I'm asking, is there a tail to that, as in a long tail extending out from those initial justifications for choosing somewhere like Cambridge?
Yeah, I mean, it really is. I mean, it's a reminder of exactly how fragile the early biotechnology industry was. Like some interviews with like Biogen leaders were like, we need to be close, in part to reassure people that if the company goes under, they can like re-enter an academic community. And I do think, I mean, one of the things that I'm trying to make sense of is it's I guess like, even if you see stability, it doesn't mean that things aren't happening. And there's this constant push and pull between the difficulty of really building biotech companies in this densely urban environment where it's deeply expensive to do work and often very inconvenient in other ways, such as like the increasing lack of parking. The Massachusetts Biotech Councils today, one of their big issues pre-pandemic was the accessibility and reliability of public transit, because traffic was so awful. But there's always people attempting to leave, but there's an equal number, if not more, either deciding they have to stay or trying to enter. And this is sort of over the horizon of the book, but very much a part of the story today, is the new set of actors that comes in that really like the idea of being within walking distance are pharmaceutical companies. And they sort of start entering in 2006. And their willingness to pay a premium is a decisive factor instead of really fueling the expansion of pedestrian scale development. On the opposite end, you see efforts such as Lab Central, which are sort of incubators for very small biotech firms, which are once again very much insisting on the need to be spatially close, as opposed to simply socially close to to a major university and to other things. So it's just very different than how either the lifestyle-based concerns of Silicon Valley or say a major pharmaceutical company initially, which in the 60s, 70s would have been interested in just not necessarily having too much exchange with other people because they were very much concerned about their own product. But sorry, yeah, so that's a brief answer, but it's always sort of a compressed push-pull. Lots of other communities are eagerly trying to get these companies to come set up, and it's just a question. It's remarkable that in that sense that more don't leave at a certain point.
Robin, I have a very quick question. First of all, thank you very much for a great presentation. It's actually intrigued me just to wait for your publication, and I hope it will be published soon, your book. But do you plan to include in your book stories about those companies which were very, very successful at the beginning and sadly we don't know about as much as about others?
I mean, that's a research challenge. Like the answer is yes, and that's another reason actually. If you'll forgive me, that the Cold Spring Harbor archives are so interesting, because you can use them just to get a glimpse of an industry that in many ways doesn't get preserved. So, for example, to pick on Sidney Brenner, he was a consultant for the Rothschilds investment bank, which wanted to invest in American biotechnology firms. They paid him for several years to go on trips around the United States looking at potentially promising companies. And his notes are in the archive, which include companies that he visited that I've tried looking up and are certainly no longer in business. So it's also a way of keeping and contextualizing the industry with a mind towards a number of these companies that that don't become the big names.
Yeah.
So sorry, yeah. So it's something I'd like to do, but it is also how do you find traces of companies and activities that don't make it to the famous prize-winning or otherwise established level that we usually would look to?
Nathaniel.
Well, hi, thanks for that, Robin. That was really great. I love hearing about this project as it comes in. You started by briefly mentioning the recombinant DNA debate in Cambridge, and at which time biotech had the predecessor had a very negative PR in Cambridge, and by the end of your talk, we have biotech having a reputation of being a beneficial alternative to this NASA structure. And so I'm wondering if you could just say a little bit about how the city and the surrounding cities navigated that, and the companies with them navigated that change in public image from dangerous, hazardous keep out to a positive business community that was not going to damage the area.
Yeah, I'm just saying, I hate to say is the subject of the article I'm currently writing up for the JHB, but it's tricky because several things have to be true at once. Like, people do remain concerned about safety, but it's interesting that they become less well represented at debates over permitting or allowing development than people who are concerned about jobs and the type of community. And so it transforms when someone like Al Vellucci, who's the mayor of Cambridge, who's very famous for being very avidly opposed, emerges as somebody who, when he's finally talking with biotechnology companies, tends to have a more euphemistic, pragmatic view. They are interested in the promise of employment. The promise of employment becomes important. But then later on, you have a new set of conflicts within the city, which is between the city administration, which often sees the tax revenue generated by these laboratories as being very important to the health of the city, versus people in the neighborhoods around biotechnology developments who are asking about questions about if there's employment generated, and the answer on that is equivocal because there's and also things such as like toxic soil brought up by construction, or the number of cars that are causing pollution, because despite the fact that people like living within walking distance of MIT, they don't actually want to live at MIT, so they're still commuting in from the suburbs. And biotechnology is sort of very much, once again, has a social geography that's different than just everything in the suburbs. And the very fact that you have more people commuting in has its own environmental impacts. So there's a number of new questions that people have about how land is supposed to be used in Cambridge, but they're sort of about the health of a city in a very different way than the recombinant DNA debate. And along with that, though, there's both a cultural and legal diminishing of the land that's being developed. So rhetorically, people say this is nothing. It was parking lots. It was a moonscape, which of course it was a moonscape designed in the process of, I guess, trying to get to the moon. If you'll forgive me. But the other part of it is, legally speaking, within urban redevelopment. This is what I got the opportunity to spend time learning about. It's designated as blighted and decadent land, which allows you, legally speaking, to do a whole number of things development-wise that are much harder to do in other parts of zoning, which I don't want to inflict that on this audience, but that sort of ends how zoners, how zoning and urban planning classify land, legally speaking, plays a big role in making this possible too, by like lowering what that land is, which is how very much it becomes also a story about American urban history and the types of cities that are being made and unmade after the Second World War.
Thank you.
So thanks again to Robin. Wonderful. And now we'll turn to our final presentation for today.